So these are the 7 things I would like to walk you through, all of which have to do with differences in one way or the other. Experimental design for field experiments, recovery of interblock information, data analysis, heritability, coefficient of determination for GLMs, and finally, spatial methods for field trials. I may not be able to tell the full story for each of those for lack of time. So just for background, I mean, as Francis was saying, my background is in plant breeding and agronomy. That's my field of application. And I just thought I'd bring a few pictures to give you an idea. This is the kind of stuff that I'm working with. Field experiments, phenotyping platforms, and well, it says field experiments, but there's also greenhouse experiments, which call for experimental design. And it's always like row and column dimensions there that are relevant for generating a design. Or in a growth chamber like this, or the smaller number of units, this time it's pots. And another one that's actually from Canberra, from ANU, with Arabidopsis. So the first is very briefly on experimental design, where the design involves blocking the control for local control of error. And the basic model that's used for analysis has a treatment effect and a block effect, so we can adjust for block differences. And the criterion that we use to find an optimal design, and that's relevant when the blocks are incomplete, how do we compose the blocks, is essentially the pairwise variance of a difference between 2 treatment effects. And averaging that, we have the average variance of a difference as the criterion to then search for an optimal or for a good design. And here's an example where there's not just 1 block factor, but 2 block factors, so rows and columns. And it's an unresolvable row column design for 25 treatments. And what you can see here is that, as highlighted by the different shading of the plots there, that some of the pairs of treatments, like the 1 and the 19, occur right next to each other a couple of times. And also you can see that 18, treatment 18 and 6, they're kind of clustered. But this is a perfectly valid row column design. If you just evaluate on efficiency in terms of the row and column blocking, it's fine, because they're in different rows and different columns, all the applications for all of the treatments, so that's fine. But practitioners don't like this. They don't like this at all. They want more even distribution of their treatment replications across the layout, and they want neighbor balance. They don't like these neighborings here, these neighbor relations. And here's one photograph to show why. This is frost damage in a field experiment with barley in Germany. And these plots here have really died of frost. And what they do in these trials, if a plot really is lost completely, they will also discount the data from the 2 neighboring plots because of neighbor effects, because the distance between plots is so low. So that's really why they are not going to accept a design like this. So with Emlyn, we tried different things, one model-based, using spatial covariance structures, and the other using a model-free approach, a more algorithmic approach, to make sure that these configurations are ruled out in the search for a good and efficient design, monitoring row column average efficiency factors. And you see that, and you could check for any of the treatments that they're well spread out, and the neighbor balance is much better. It's not perfect, but it's certainly better. That's the kind of work we've been doing. One of the things we've been doing for experimental design. This is non-resolvable, also works for resolvable designs and partially replicated designs, which are very popular in plant breeding. And this is just to sketch that we are using one model-free approach that really monitors the neighbor configurations around each of the plots, and then make sure that in the neighborhood you don't have these undesirable configurations. Yeah, of course, that's a restriction on randomization. And there's many different ways you could do this. And one is really using a very specific spatial model that imposes a specific covariance structure. But then you need to make some very strong assumptions about what the model is going to be that you want to use in the analysis. And that's hard to tell before you start the trial. So there's really a lot of options there, and it's not clear that you could do, that you can really use exactly the same model that you use for generating a design model-based. So our model-free approach has the advantage that it doesn't make any of those assumptions, and we consider that an advantage. So second item is recovery of interblock information, the same model, same idea. And then when you model the blocks as random, you can recover the interblock information. Essentially, that's the information that's contained in the sums across the observations in the block, as opposed to the differences within block. That's the intra-block information. So modeling blocks as random provides a little extra information that improves the efficiency in terms of the average variance of a difference. And here's a few slides now that give a more general setup that will be relevant in that context. We'll generally be dealing with a linear mixed model of this form. And then both the fixed effect and the random effects can be partitioned. And there may be effects that are of primary interest, both in the fixed and the random effects, and other effects that are not of interest. And the block effects are kind of nuisance effects. Of course, we're interested to control for them, but then we want to get rid of them. And we can then also partition the solution that we obtain for the fixed effects as well as for the random effects. And once we've done that, we could actually consider correcting the data, subtracting the effects that we're not interested in. And people actually do that. They do that in animal breeding. I've seen it several times. And when I first saw it, I says, hey, this can't be right. You can't just do that. If we knew those two effects, we could just subtract. And then we'd have a corrected observation. And then we could fit that reduced model here. And everything would be fine. But the problem is we need to estimate those effects. And so what we need to do is then plug in the blue and the blob for the nuisance effect, subtract that. And then we have this corrected value, the properties of which, of course, are different than when beta and beta 2 and U2 were known. But then you can show that you can analyze this model, pretending that the errors in that model here really behave as the errors in the original full model. What this will do is it will produce point estimates that are correct, both for the fixed and the random effects of interest, but they will not lend themselves for further statistical inference because, for example, the variance of the estimator of the fixed effect, the blue with beta 1, obtained in that way, is not equal to the naive estimator that you would obtain, pretending that the variance structure is really the same as in the original model. So what would be required to fix that would actually be some kind of meta-analytic approach where you work out what is the variance-covariance matrix really of my adjusted values if you want to go down that route. And then consider that in the next step of the analysis. I don't think that's really the way the data should be analyzed, but it does provide an interesting view, I think, on the recovery of interblock information. So what can be shown is that if your variance for the random effect for blocks, assuming U2 is the block effect, if that goes to infinity, then it's known that the blobs behave a lot like the blues. And so there's really kind of a maximal correction for block effects. And so there's really no interblock information anymore because you have such a strong correction on your original data. You're subtracting large effects when there are large block effects. Conversely, if the variance of that effect goes to 0, then the blobs will be shrunk very close to 0. So the actual correction that you're applying in obtaining those corrected data is minimal up to really 0. And then you've got no correction. So all your information is interblock information. And I think that's interesting, provides interesting insight into how the recovery of interblock information works. And I've used that in teaching to agronomists with hopefully some success. Another application that's actually quite common too is to have a spatial trend model, for example, using P-splines. That's very popular in field trials, and I'll talk about that a little later as well. And then the nuisance effect would be a spatial trend effect. And then you get these nice figures that show you, okay, this is your spatial trend in your mixed model, and this is implemented in the mixed model framework, of course. And here's your signal that you obtain after subtracting the trend effect from your observed data. And that's kind of nice. So you see, okay, you know that there's trend. Can I get rid of that by just subtracting it? And then proceed as if the trend had never been there. That's actually what people do. But then you've got the same problem that you're not getting the downstream inference correct. Right. Continuing with recovery of information. So when the block variance is estimated to be 0 in a model where you fit blocks as random, then effectively the block effect is removed from the model. And as I've just shown, then there's going to be no correction for any spatial trend. But we kind of know that in the field, there's always some heterogeneity, and we kind of expect that always some heterogeneity can be captured by that random block effect. But then occasionally, the variance is estimated to be 0, especially when the number of blocks isn't that large. So we consider the idea of using historical data to estimate and inform a prior distribution for the block variance, and then use that in the analysis of new trials, using the historical data to build a prior, and then obtain a modified estimate of the block variance and use that in the analysis for new trials. We did that using Polish data, using historical data, and the designs were alpha designs, where there's complete replicates and incomplete blocks nested within complete replicates. And this is for 24 treatments taken from this book. And so the model then has a fixed effect for replicates, here, and it's got a random effect for blocks and residual error terms, so 3 random effects. And this is for the historical data, a histogram showing the estimates we obtained for the replicate variance and for the block variance, and the percentages of zeros are pretty large. So it's a real issue, mainly for this one. I mean, this one doesn't have the role really in the recovery of information, but this one does. So the idea that we used was, well, of course, you could try and build a prior here, but then you're dealing with a spike at 0, which is kind of difficult to deal with. So what we did instead is take a classical analysis of variance approach and use the sums of squares for replicates, blocks, and error, and worked out the expected values of the mean squares or sums of squares, and used that to build a distribution across the trials for the block, replicate, and error variances, assuming that the sums of squares are conditionally independent, which they are, and then using a model for log variances, assuming, for example, a multivariate normal model. But we also tried some other models, but that's just one example, and then did empirical Bayes or a fully Bayesian analysis. And here's just one set of results where we compared REML to those hierarchical models. And so REML is really the standard approach where you get all those zeros. And our criterion here is the mean squared error of a difference. And you can see that there's really a substantial gain here, especially when the block sizes are very small. I mean, this was simulated for 100 treatment, and this was simulated for just 6 treatments. That's really small, not so commonly used in practice. But just to see how much gain you potentially get. Of course, the larger the trials get and the larger the blocks get, the less gain there is. But it's essentially, the point is that that gain is essentially for free. If you just use your historical data and have them basically inform your prior and updating your prior in every year, you get a little extra precision basically for free. And we thought that was nice. On to the next item that has to do with differences. And this is meta-analysis. So here's an example, incidence of diabetes with treatment with various drugs. The response is binomial. There are 6 treatments and 21 studies. and the data is very incomplete. So this table is treatments by trials. And so you see there's a lot of empty cells, and that's very common. And then the method to analyze this type of data in the meta-analysis is called network meta-analysis, because it's not just pairwise. I mean, some of the studies are pairwise, but you're not only looking at 2 treatments overall, you have 6 treatments across all of those, and you want to do all the pairwise comparisons. So there's a lot of methods and there's a lot of work there done in this direction. And there are 2 different approaches to do this. And I'll talk about those in a minute. But the key issue there is that there's direct comparisons and there's indirect comparisons for the treatment. So if I've got a study where the 2 treatments are present, I've got a direct comparison right there. But for other comparisons, I need to really have indirect comparisons across the studies using maybe a placebo treatment that's present in many of the trials as a reference. So, and that's actually very closely related to incomplete block designs. If you just think of the trial as an incomplete block of your treatments. And that analogy can be exploited in setting up methods for analysis. So, yeah, so this is just illustrating this idea of direct versus indirect comparison. So if I've got a direct comparison A versus C, and I've got another direct comparison of B versus C, computing the difference, I get a comparison between A and B. That's the kind of indirect comparison that's exploited in network meta-analysis, and there's very sophisticated ways of displaying that, of exploring how many indirect comparisons do we have, what's the connectivity throughout the network of trials. And most of the methods of analysis are really contrast-based. So they really work directly on differences between treatments as estimated in the individual trials. So that's where the differences come. And then the whole modeling approach is set up for the contrasts among a treatment with the baseline treatment. So for each study, there needs to be a baseline treatment against which every other treatment is compared. That's how it works. And that's by far the most common approach used in network meta-analysis, especially in the medical field. And the other approach is called arm-based, where the model is not for differences, but for the treatment means directly. Treatment means, log odds, log risk, and so forth. And the model is essentially a two-way ANOVA model. So the contrast-based model, as I said, works directly on the contrast relative to baseline. So this is assuming a normal distribution. And then this variance tau squared, that's a variance for heterogeneity that's used in meta-analysis, heterogeneity between the trials. So we can't expect the effect to be exactly the same in each and every trial. And then these are the outcome variances of the summary statistics computed for the treatments in the different trials. So that's the contrast-based formulation. This is formulated for summary statistics. You can also do this for individual patient data or individual person data. And then the model looks a bit bigger. And this is the arm-based model, where essentially you have this linear predictor, which has all the fixed effects. So here the study effect, the study main effect, and here's the treatment main effect. And the interaction effect really is encapsulated here in the variance for heterogeneity. Alternatively, I could also write it as a mixed model with fixed effects, those two plus a random interaction effect, which has this variance, which I also denoted as tau squared over 2 to make it correspond to the tau squared in the contrast-based formulation. So those are the two approaches. And I find this approach much more natural coming from agriculture, where there's these incomplete block designs and this analogy with network meta-analysis. Whereas most of the software is really focused on a contrast-based approach. And some of the things that are done in that context really look a bit complicated, unnecessarily complicated, whereas this is just the two-way ANOVA. I can fit that easily in any mixed model package. That's the main idea there. And then the recovery of information thing comes in again. So if we continue with that analogy, trial is behaves like a block, basically. If trial effects are fixed, then we only use the within trial information. And some people insist that in meta-analysis, you should only be using that information, because that's the only information that's protected by randomization. I mean, that's the analogy with incomplete blocks, but it only goes so far. I mean, we're not really designing this as a trial, and so there is no randomization step among the studies that would justify a random blocks assumption. So that's why people insist we shouldn't exploit that information. So you can achieve that by just taking the trial main effect as fixed. Right, and if you do that, you actually get complete agreement with between the contrast-based and the arm-based approach in the case where you are modeling summary statistics and you're using REML. We've shown it in a paper in 2012. When you analyze individual patient data, it's no longer exactly the same. There are differences, slight differences, but still the models really are related together one to one. There is issues with estimating the variance components when you have an individual patient data kind of setting. And so there's issues with degrees of freedom, and we've done some work on estimating the variance components in a case where we use an arm-based formulation of the model. And but if you can bring in REML or REML-like estimators of the variance components, it's working quite well. Okay, then and then when we model trial effects as random, some people do that in meta-analysis as well. Yeah, then you're recovering that information. That's a little extra, actually quite a bit extra, because the blocks are quite small. It's only block sizes 2 and 3, so there's a tendency. Well, it's attractive to do that on the surface, but then we should probably really keep this advice here. Yeah. Well, where does this come from? Meta-analysis, just a little side comment there. It really goes back to this paper by Yates and Cochrane, I think, because they did series of experiments in agriculture, but the methods there are really the same methods that can be used in meta-analysis and are used in meta-analysis today, especially when you use this arm-based approach. And in that paper, of course, they also have all these weighted means ideas, which are, of course, key in meta-analysis, because you've got those outcome variances, which then give different weight to different observations in the data that you extract from the study. So it really all goes back to this paper, I'd like to think. Right. My next item is heritability. So heritability is a measure that plant breeders like a lot to characterize how informative is my trial, what's my genetic gain going to be, and so forth. And in plant breeding, it's usually multi-environment trials for which this is relevant. And the model is, again, this two-way model where the main effects are for genotypes and sites or environments, and then the genotype-environment interaction. And this is assuming you've got randomized complete blocks, which is still being used. I'm using it here because the equation is going to be simple. Of course, I would be advocating incomplete blocks and a residual plot error. So based on that, you can work out what the heritability is. Here's the general definition: is genetic variance over phenotypic variance. But what's your phenotypic variance? And what is your phenotype in this kind of setup? Because as opposed to animal breeding, here we can replicate the same genotype. We can see it in multiple environments. That's the big advantage. So is it a single plot observation? Is it a genotype mean in a single trial? Is it a genotype mean in a multi-environment trial? Is it a block? And how do we estimate the phenotypic variance in all of these cases? Well, I'll be focusing only on this case because in my mind, the phenotype is always the blue. I can always cast it as a blue, even if in the end I want the blob. I can get the blob from the blue if I want to, as a final polishing step, as it were. So my phenotype is always going to be this. And that's actually the phenotype that's considered in a very basic equation for the phenotypic variance. So this is the usual equation that you see in broad sense heritability equations in the denominator. And in the numerator, there's the genetic variance. So genetic variance over this, that's your heritability. So, okay, that's the phenotypic variance. It involves the genetic variance. Okay, it involves the interaction. Okay, it involves the plot error. Okay. So what about this and this? This is the model average for my genotype. So this is my blue for a genotype averaged across sites and replications. And what happened to these? They also have a variance, don't they? So why are they not featuring here? My view of this, and you know, I get this question every once in a while also from plant breeders. Sometimes they just plug those in and sort of say, that's my phenotypic variance. And I'm saying, no, those shouldn't be in there. Because to me, this quantity here, I don't really consider this as the variance of the mean, especially when site is a random factor. I forgot to point that out. We're breeding for a target population of environments, and so the sites are regarded and obtained, hopefully, as a random sample representing that target population of environments. So site is a random factor, and so both of these effects should be modeled as random. But they don't feature in this term here. And my view on this is it's because it's half the variance of a difference between two genotypes. And that does make some sense. Of course, then what is this? Well, this is half the variance of a difference between two genotypes. So the variance of a difference between two genotypes would be 2 times sigma g squared if you assume genetic effects are independent, and that's half of that. Yeah, so, but then data look like this. So each of these is a lattice design. These are six locations, and there's lots of lattice designs placed right next to each other. They're connected using checks. So this is very unbalanced. So what do you do in a case like that? Well, continuing with that idea that it's really about variance of a difference. Well, why variance of a difference? I also forgot to point that out. Breeders are selecting, so they're comparing genotypes. So they really are interested in comparing the genotypes. So that's why the variance of a difference is relevant. So this leads to the idea that we could define a heritability for pairs of genotypes, for individual pairs of genotypes. And then say this is the it's the variance of a difference among my two genetic effects divided by the variance of the estimate of those two genetic effects based on blue, for example. If it's blue, yeah, then it turns out to be this equation. And then we could average that across all the pairs to give us this. If you do the harmonic mean here, you obtain this equation, and we propose that as a measure of heritability. So here's your genetic variance over genetic variance times half the variance of a difference among your blues. You can do the same with block. And then you do that for a pair, you obtain this, you average that, you get this expression here, which has been proposed. Both of those work with, assume that the genotypes are independent, the genetic effects. But then when you use marker data or pedigree data, and that's what plant breeders and animal breeders often do, then of course there's covariances between the effects, and then one needs to take those into account. And there may also be heterogeneity of variance. So that leads to really one pairwise heritability for every pair. And if you've got a balanced, very simple setup, completely randomized design, then every pair has the same heritability. But oftentimes there's some variability. I mean, this is an alpha lattice design. It looks like a big difference, but if you look at the values, there's not a big difference there because alpha design is a good design. And so these are really very close. But then when you include kinship, you get something like this. You could still compute the average, but it's probably also good to look at the individual pairs and consider what that means, what those pairs really tell us. And what they tell us about the design. If I've got a very wide spread among my pairwise heritabilities, maybe I need to reconsider the design that I'm using and try to make sure that the pairwise heritabilities are more similar. And really, most of the time, there's some form of incomplete blocking, there's imbalance in the data. And so it's much more common to really end up with this situation rather than having really the exact same value for all pairs of genotypes in heritability. Right, continuing with my list. Am I doing on time? Yeah, but okay. So coefficient of determination, I mean, here I can't really tell the whole story. There's a lot of work on coefficient, a lot of interest in coefficient of determination of measuring goodness of fit in GLMs. And many of you I know are also working on GLMs and GLMs. So, and here's one that's particularly popular. And there's a nice R package. People use that a lot. And yeah, we're not so, this is nice. I won't describe the details, but one issue I have with this approach is it's basically when you measure coefficient of determination, you need a metric for variance, for total variance in the data. And if you've got a linear model, your data is independent. That's just your residual variance could essentially just be used. But when you've got random effects, then things are a bit more complicated. In particular, there's correlations between your observations because of the random effects. And the approach that's taken here is to ignore those correlations and basically just look at the diagonal of the variance-covariance matrix of the data, but ignoring the covariance. So here's an idea how the covariances could be brought in. And so very, very generally, a data vector with the expected value and some variance covariance structure. Of course, the expected value depends on the fixed effects in the mixed model, and the variance covariance depends on the random effects. And then if we use the semi-variance, then we are accounting for the covariance. So using the semi-variance is a natural way to bring in the covariance into the equation. And then average that across all the pairs in the data set. Provides a measure of total variance. And this is actually a discrete version of what's used in geostatistics, where you use this double integral to measure the variance within a region, taking into account the spatial variance. So, this could be computed for a linear mixed model that only has an intercept, and it can also be computed for a linear mixed model that has a couple of fixed effects, and then the difference between the two will be the explained variance. And based on that, a coefficient of determination can be defined. So I'm not giving equations for coefficient of determination. I'm really focusing on how do we measure total variance? What's our metric there? But then this average semi-variance only captures the total variance in the random effects part. If we want to bring in the fixed effects part as well, then it's useful to look at the expected value of this squared difference in half of that, expected semi-squared difference. So I'm calling that ESSD, expected semi-squared difference. And that can be worked out for every pair, and then averaged across the whole data set, and then added on to the average semi-variance. So this is the average semi-squared bias, what I call the average semi-squared bias. It depends really on the use, on the fixed effects, and this depends on the random effects. So one way to then define a coefficient of determination is to take this as your numerator and take the sum of the two as a denominator and call it a coefficient of determination. And we can show that if you use an unbiased estimator here, then the whole thing really is identical to the adjusted R-squared in a linear model. But then it can be extended to linear mixed models, can also be extended to generalized linear mixed models. There's challenges there and extra things that need to be considered because the randomness occurs at two levels, at the observed level and on the linear predictor scale. So one needs to project the variability from one to the other. One needs to settle for one of the two where to assess R-squared. So that's really challenging. But so I just wanted to bring this idea that semi-variance, variance of a difference, could also be used to define coefficient of determination. Very similar in spirit as my interpretation of heritability. One can also show that this measure is actually can be related to a sample variance. If we consider computing a sample variance across our data, ignoring the mixed model. This is just my sample variance. And then we work out the expected value of that under the assumed linear mixed model. We can show that the expected value, this is actually our measure of total variance, which is kind of nice. Yeah. And now to my final set of slides on spatial modeling for field trials. So the very early methods looked at neighboring plots and said, okay, maybe the two neighboring plots can tell me something about the central plot, and maybe I can use that for making some kind of adjustment for fertility differences. So the idea is to look at the observed value in that plot, subtracting the average of the two neighbors. And there's a lot of variations and modifications of that very basic idea that's going back to Papadakis. And this idea can actually be shown, if you use this kind of correction, it can actually be shown to be essentially, well, at least very closely related and in some ways equivalent to the linear variance model, which again Emin proposed. And this says the covariance between plots decays linearly with spatial distance. That's the linear variance model. And that means that this kind of very old idea, nearest neighbor analysis, can actually be implemented very easily in a linear mixed model package if you can specify a linear variance covariance structure between your plots. Can also be related to another type of model, state space models or random walk models. So under a random walk model, the pairwise difference between two neighboring plots in terms of the trend values on those plots, if that's assumed to be IID normal with constant variance, then we have a first order random walk. And that model is also equivalent to the linear variance model. So they're all really very closely related. Or you can do a second order random walk, which some people have proposed for field trials as well. And you can also regard the AR1 model as a special case of a state space model, where instead of, so you basically have an update. You can also rewrite it and say, okay, my trend on plot 2 is the value on plot 1 plus a random perturbation. That's a random draw from a normal distribution. But instead of doing that, you can maybe have this dampening factor. You can call it the dampening factor between 0 and 1. And that's also commonly used in state space modeling for time series, of course. But this is really the AR1 model. It's equivalent to the AR1 model. And then there's also a connection with P-splines, which have become very popular for field trials. So, yeah, I'm trying to sketch the idea in the context of field trials. So there's going to be random effects that will be used to model the trend values for the plots. But now there's going to be potentially several of those random variables which are combined linearly to form a prediction or form a trend value for an individual plot. So each plot trend value is a linear combination of those random effects. And those random effects are assumed to be normal, can be assumed to be normal, and there can be a penalty in the P-spline framework, but that can be translated into a mixed model where we just say these are random effects, they are random draws from a normal distribution, and then the penalty is in terms of differences, either first differences or second differences. And then these coefficients can be taken from B-spline basis function, which have some nice properties. And this is just an example of a first degree B-spline basis and a cubical B-spline basis. And here's an example how the fitted trend looks like when you use, for instance, a third degree P-spline. And the red dots are trends you obtain when you use a linear variance model. So these are different models now, but what they do in terms of predicting the trend or modeling the trend in a field trial, this is just the spatial ordering plots is also quite similar. But then the connection can be closer. If you look at special cases of P-splines, that's going to be my and a final point. So this is just a few examples. A cubical B-spline, if you have 5 plots and 7 knots, then this is what your B-spline basis matrix looks like. And then your trend values is this linear combination for the first, this linear combination for the second, and so forth. So it's combining several of those random variables U. And this is a third degree with just 5 knots. But then when you use a first degree B-spline basis, it's still a P-spline then, and you use 5 knots because you've got 5 plots, then your B-spline basis is just the identity. And then when you use first differences as your penalty, this actually is essentially the same again as the linear variance. So that puts all these earlier approaches into context with the P-spline approach, which has become very popular and initially looked like a completely different animal. But it is really closely linked to what's been done earlier. This is just for one dimension. Of course, you can extend that. You need to extend it to 2 dimensions because you've got rows and columns in the field layout. And then you can use a direct Kronecker product, both for the B-spline basis and also for the penalty, which is sketched here. So this is a differencing matrix that implements the penalty, either first differences or second differences. Both, and you can do it for the rows and you can do it for the columns, and then multiply those together using the Kronecker product, and use that to define your penalty matrix. This is your penalty term, and use a penalty matrix. And you've got a parameter in that penalty in the spline framework, but you could interpret that as the inverse of a variance of a random effect and fit that as a mixed mode. That's how it works. And here's just an example of the nice plots you get out. So this is the observed data, and this is the fitted trend. So that's what the P-spline does. Or a P-spline does in this case. And we've tried it on a few data sets. 2 data sets are shown here. This is kind of small. May not be able to read it from the back. But we started with a baseline model, which is reflecting the randomization structure, rows and columns, or just incomplete blocks and complete replicates. And then added on various spatial components, classical ones like the AR1 model or the linear variance model. And we looked at AIC for goodness of fit. Of course, the problem with P-splines is there's so many knobs to turn, and that's kind of a risk there as well. We did that too because we just wanted to explore it. But then it turned out that one model that's quite simple, really, because it's a first degree B-spline basis, and that's the one that's really so close to linear variance, as I said, and then places the knots exactly at the plots. Does pretty well. Actually, it was the best fit among models in terms of AIC. If you use first differences, this is using first differences. We also tried a couple of things, and there was even more things to try there and to take care of. But none of those did better than when using first differences, interestingly. So that's, I don't want you to read any of that. It's just a reminder to say that we also tried second difference. Yeah, so P-splines are very versatile option for spatial modeling in field trials. There's many knobs to turn, which is an advantage. But first differences work quite well, although second differences are more commonly used currently. Right, and yeah, we always start with a randomization-based model for analysis, and then just check is spatial modeling, if you add it on as a modeling component, is it improving the fit? But we always want to have the randomization-based analysis as a fallback if the spatial modeling doesn't really work. And of course, you know, a sophisticated spatial model is not analysis is not a substitute for good experimental design. I don't have to tell it to you guys, but a lot of people seem to think that you have these sophisticated methods that can correct away your trend. That's cool. We just do completely randomized design, and then we correct for any trend that's there, and then we're going to be very powerful. That's a better idea, as David Cox reminded us. We put him on the slides because he, I think, gave the NIPS lecture in 1977. Is that right? Yeah. Yeah, I don't really have a summary. I've struggled to summarize them, other than saying this is all about differences. And there are some connections there, you'll hopefully agree. And yeah, with that, I'll be done.
Very much, are there any questions, first of all, from the audience you would like to ask? Yes. Yeah, sure.
So regarding your neighborhood design, have you seen this type of design used outside of agriculture?
So the question was basically with the neighborhood designs, has there been any applications outside of agriculture?
I don't think I've seen any. I mean, have you seen any application outside of agriculture of this particular way of doing neighbor balance? I haven't seen any applications. But it's a fairly recent proposal anyway.
Did you see any potential? I'm just wondering whether this can be generalized outside somehow, where maybe somewhere else medical say can benefit from it.
If you have this spatial layout with rows and columns, yes. But I'm trying to think of an application in the medical field where you have that.
Yeah, I struggle.
Well, maybe those 96 well plates that I used so much for samples. There's rows and columns, obviously. I haven't seen applications of row. There may be applications. It would be very interesting to do row column designs on those well plates to begin with, and then that could be useful there. Yeah.
I have heard anecdotally that there's an effect that is not corrected for in the well plates. That people sort of know that they should randomize, but they don't figure it.
Yeah, yeah.
But that's anecdotal.
Sorry,
no.
Any other? I don't think there's any questions online at the moment. We do have quite a bit of time. So, does anybody would like to ask any other questions? Yep.
What do you have sort of particular packages that you always go to for fitting your mixed text models?
I'm using SAS a lot. Actually, that's all I use because I was raised on SAS. Everyone else in my group uses ASReml and LME4 and all those other packages. So I always ask them for help. Or I ask them to, can you do the same thing in R? And many times they come back and say, I don't know. And they tried many things. And sometimes you really can't get the same thing. One of the things that I've been struggling to find, maybe some of you guys can tell me later, residual or pseudo likelihood approach for fitting GLMMs. That's the default method in ProgMix, in ProgLimmix and SAS. I've been struggling to find that in now. There's many methods, of course, to fit GLMMs, but I couldn't find that method. Yeah, but I mean, any good mixed model package can be used to do this, of course. Yeah.
Okay. Any other final questions before we wrap up? Okay, so if not, thank you very much, Hans-Peter. Thank Hans-Peter again.